so I don't forget. So we are going to go through photosynthesis and cellular respiration and try to highlight some similarities as we go. You have your exam that will open up today. Let me share our screen. So on Blackboard, right, I put an announcement out the other day, and it reminded you about the exam. And then the flip grids, I'm going to start removing the flip grids that you no longer can do to kind of keep yourself organized. Not an attempt to do anything other than that, right? And it's really fun watching them. Hopefully you're getting something out of them as you talk about them. And I noticed that you guys are watching each other's, and that should be really helpful. Just hearing it all sorts of different ways is great. So then you find the exams here in the exam tab, right? You click it, and it will eventually appear to you guys today. And it'll be, very similar to the other one, just a couple minutes less time. But when I looked at all the analytics, you all finished well in advance of the time. So we should be just fine. So questions on the exam before we get rolling in connecting the content and running through photosynthesis and cellular respiration again. Jillian.
I actually don't have a question about the exam, but for the flip grids, the 6 that we have to do, does the introduction one count towards that, or is it 6 and also the introduction one?
Nope, it's 6 including the introduction one.
Okay, thank you.
Yep, good clarification. So then, and thank you for talking, that's delightful. So if you go into documents for class and you read the flip grid, you sometimes have to reread things, right? I have to do it all the time. And so it kind of goes through that the intro is one that you should have done and that you have to do 5 more in addition. But keep the questions coming with that because that will be helpful, right? Because I don't know if you don't understand it until you ask me. More questions? Yeah. So the content from exam 1 will be on exam 2. Like I'm not going to ask you, is water polar? But the fact that water is polar and then that it creates hypertonic and hypotonic situations, that's all applicable. So all the language continues so that the content is built upon. So no longer will I define what hydrolysis is. You need to know what it is in context. Does that make sense? Yeah, so not explicit questions, but all that information is completely foundational to this stuff. More questions? Yep, can you still turn in the flip grids after class tonight? Yes, if you look at the due dates on the flip grid, they're due at midnight tonight. So the point of the flip grids is to engage in the material for that upcoming exam. That's why they wouldn't be due after the exam because that's worthless. That's just jumping through a hoop at that point, right? And so they're supposed to just get you to think about saying these things out loud. When is the exam due? It's in the announcement. I'm not trying to be super cagey, but there's lots of places you can find the information. It's the same that it was last time. It's in the announcement and also will be linked up in the exam. This is where, again, let me plug the Student Academic Success Center. Learning to keep track of stuff in a better way is a skill that has to be harvested. And so I have to do it too, for Pete's sakes. So, right, it's due at 11:59 p.m. on Friday, just like it was last time. That does not mean it started at that time. That means it's done at that time. More questions like that? And I'll always answer your questions. I just also want to show you where you can find it. Okay. So we're going to go through, I think I uploaded the same device. I did. Hang on, I'm going to add another file. I need to put them as PDF so that it doesn't change the formatting. Okay, so this is on Blackboard in our Module 2, Exam 2 materials. I added these last couple PowerPoints for us. And one is the blank pictures. One is those blank pictures filled in. And then one is this kind of tie together piece. And so we're going to flow through this first, and then we'll go out and actually draw through these processes again, okay? So we'll do kind of both. So I think it's important to see how these two things relate. And as we go through, it's going to kind of be defining these terms. So what I need from you is questions. If a sentence doesn't make sense, flag it for me, and so that we can kind of make sure we're understanding what that exactly means. Because hopefully you're at the point where you've got the words down. Now it's just trying to see exactly how they mean in context. So photosynthesis is an endergonic reaction. What does endergonic reaction mean? What does it need an input of? Energy, right? So this needs energy. Photosynthesis needs energy because it's against the second law of thermodynamics. There's not energy available. It's not free. So chloroplasts split water into hydrogen and oxygen. Where does the oxygen go? What happens to the oxygen? It's released, right? It's released. The plant cell doesn't directly use it for photosynthesis. It uses it for cellular respiration, but it's released from the chloroplast. Oh no, do you hear staticky noise, Stephanie? Anybody else hear staticky noise? Yes, 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 I do. Yeah, so that's a yes. Just when you talk, I think. Well, isn't that convenient? Like just when you breathe. Okay, let me try to move the in-thing mic. We haven't had this problem prior. Is that better? Keep talking. Is that better? Is it better now? You all respond. No. No. Wish you guys could see my chat. Oh, you can see the chat window. Okay, is that better now? Has that been clearer now that I changed? It's not that loud for me, not really noticeable. It's still not better. Not that bad. It's not super loud to me. I'm repeating these so that you can continue to hear my voice as I mess with the volume up here. Is it better? It's fine. Is it getting better? Okay. Okay, keep telling me if it's junky, because I can mess with it a little bit more with my very limited knowledge of how to mess with it, full disclosure, right? Okay. So, it's a byproduct, right? Oxygen is going to be used in cellular respiration, but in terms of photosynthesis in the chloroplast, it's not needed. And so it can just leave the chloroplast at that point. And then what happens to the hydrogen? Remember, it stays in there. And it stays in there because it's going to get pumped against its concentration gradient. We need to figure out what's in there, like what is that space, and then how is it moving. So those electrons or those hydrogens are eventually going to get incorporated into the sugar molecule with the electrons doing that. And we're going to look at that flow again. So photosynthesis is a redox process in which water is oxidized and carbon dioxide is reduced. So remember, reducing has the energy. So these 6 carbon dioxides are going to come in, and the energy of the sun is going to be transferred into our 2 molecules of molecular energy. Okay, so this energy is going to get transferred. Carbon dioxide becomes reduced from the application of that energy. And it gets reduced into C6H12O6, which is glucose. Now, we know that the Kelvin cycle actually starts with that little G3P, which is a 3-carbon simple sugar. 2 of these essentially kind of link together to become glucose not long after the Kelvin cycle. Water becomes oxidized, so that's that water being split. This oxygen is a byproduct, it's a waste product, it's released. So all the oxygen you breathe comes from water splitting in a plant cell. So when people think about plants being the lungs of the earth, if you think about the rainforest really being the lungs of the earth, this is why, right? Plants like that turgor pressure, they like to be hypotonic in their central vacuoles. And so they're constantly splitting water, and they don't need the oxygen directly for photosynthesis, so a lot of it is released. And that contributes to the oxygen in our environment. It becomes oxidized like this, right? And the hydrogens then are being pumped. Some will be incorporated to make the, to add those hydrogens into a glucose, because carbon dioxide doesn't have a hydrogen. So the hydrogens have to come from somewhere. They are coming from the water. So let's look at how this works again within the whole cell. So if we think about why do we have to learn cell parts, some of it is right here. So let me zoom in a little bit. I know when I zoom, you don't see it the same way. So photosynthesis is happening in the chloroplast, right? The chloroplast is behaving in a way that creates energy, but it's working with all the other dynamics that we've worked with this whole semester so far. So carbon dioxide is coming through the membrane of the plant. And so there is a cell wall that it has to deal with, but it comes through a plasma membrane as well. So that would just be passive transport. Oxygen is coming out via passive transport. And so that cell membrane right here, this is a phospholipid bilayer. Water is coming in via osmosis. And so it has a channel protein that's going to allow it to go through that tricky chemistry of the plasma membrane, because we have those large hydrophobic tails. And so that channel protein is a specific protein. So where does all that protein come from, right? It comes from the flow of genetic material. And this is what we're going to start moving to into our next chunk of information. We're going to start looking at how do we produce proteins, how is DNA replicated. And so remember, in the nucleus is the DNA. And from there, mRNA is going to leave. It's an RNA, and it's going to find a ribosome. It's going to find a ribosome in some capacity. That ribosome is the site of protein synthesis. That protein is going to be specifically peptide bonded together. It's going to be specific amino acids peptide bonded together. This needs energy too, just so we know as we start to look at cellular respiration's connection. But that particular protein, if it is the protein that is the channel protein called aquaporin, that allows for water to get across the membrane, because it's a small molecule, but it needs help to get across that membrane, then it's going to move through a vesicle, it's going to get finished packaging in the Golgi apparatus, go out to a vesicle, and that particular protein will get delivered to where it needs to be in the plasma membrane to help water come in, to help facilitate that osmosis process. So if we think about how everything connects, that's how, right? Then carbon dioxide is coming in, right? The energy transformation is transforming sunlight of energy that's hitting each one of these thylakoids. So these stacks in here, those are the thylakoids, and that's where the light-dependent reaction happens. The light-dependent reaction transfers sunlight into our usable energy of ATP and NADPH. That then is able to help fix carbon dioxide, right? We help reduce it down. It fixes it into the Kelvin cycle with the help of Rubisco, and then we reduce 3 carbon dioxide molecules into our simple sugar of G3P. Now, that would be considered the organic molecule. That organic molecule is going to go over to the mitochondria in the plant cell. Plant cells are eukaryotic cells, so they have a mitochondria as well. That mitochondria is going to use oxygen that's coming from the chloroplast. Okay, so not every ounce of oxygen leaves. It goes into the mitochondria as well. And then that organic molecule is going to go through an exergonic reaction, right? So a largely catabolic pathway, and we're going to release the energy that the plant just made in photosynthesis. And then we're going to have carbon dioxide leave the mitochondria. Some of it might go right to the chloroplast, and some might leave. But we've made ATP. That ATP now can go into the cell. Notice how large the spacule is, just full of water, full of water. But now this ATP can do things like power protein production, right? So linking together amino acids into a polypeptide, we need energy, and that ATP can go do that. Those are close-ups of it. What I look at the close-up is right here. So look at how the plant exchanges things. And so as carbon dioxide comes in and as water comes in, both via diffusion, water's coming in via osmosis, a facilitated diffusion, but they're both passive transport. And some oxygen is leaving via passive transport because small molecules can go across the membrane relatively easily. It's those large and charged ones that have the real big problem. Notice they come in, and then the chloroplast uses those molecules. Both of them are inorganic, and they turn them into organic molecules, right? We take inorganic substances, and by the magic of chemistry, they become these organic molecules. Some of the oxygen that's leaving the chloroplast can go into the mitochondria. Okay, that organic molecule is going to go into the mitochondria, and then we're going to do that catabolic pathway of cellular respiration. It's going to take the energy that originated from the photons of sunlight, and now it's in the organic molecule, and now the mitochondria can release that energy, put it into electron transport carriers, which then it's going to bring down to its oxidative phosphorylation stage to release that energy by oxidizing those reduced molecules, and it's going to produce ATP, right? ATP is going to be produced. The carbon dioxide that gets released in the pyruvate dehydrogenase complex and the in the citric acid cycle, it can be used to go right back into the chloroplast for photosynthesis. So if we think about just the general theme that we've been thinking about this whole semester, that everything is endlessly recyclable, and the fact that we have so few elements that we work with, but we have such complex processes, it's because living things are amazingly talented at taking these elements and kind of reworking them for anything they need them to do. And so the fact that carbon dioxide, oxygen, and water are involved in these basic energetic processes is really important because they can slush in between all sorts of ways to keep the metabolic processes going. Right? So these are called autotrophs because they make energy and then they use that energy they make. We don't have a chloroplast, so we're just doing this and we have to eat this, right? We have to eat those organic molecules, but then the outputs are the same. So when you go to the study by chapters and you've been watching the BioFlex animations, they highlight these kind of pictures. They highlight more of a flow of the cell, and it's different than the walkthrough animations. The walkthrough figure animations are going to be walking through kind of the more cartoon figures. The BioFlex looks at it more of how it molecularly actually looks like. Just so you know the difference, you might like one or the other. As you go, they are oftentimes really nice to look at both of them. So then aerobic cellular respiration is organic reaction. What does exergonic mean again? How is exergonic different than endergonic? Yep, it releases energy. Yep, you got it, Jesse. It releases energy instead of requiring it. That's one thing. If you've done the flip grid, you're kind of comparing the two major metabolic pathways of anabolic and catabolic. So many of you included all the language that kind of is underneath both of those terms. And that's a really great thing to try to keep separate because once you kind of know the groupings, it helps you to kind of, chunk things one way or the other. So this is the process too. So we have reduced and oxidized molecules. Glucose is oxidized. So remember, glucose is losing. This has available energy. Glucose has the energy. It's free. It's working within the second law of thermodynamics. So as the glucose is oxidized, we are harvesting the energy in those electron transport molecules, right? We're taking those electrons and shuffling them to the last step. And then water is reduced to produce ATP. So water is reduced from your oxygen that you breathe. So as we take those electron transport carriers that are reduced molecules, they have the energy, so they have an electron and a hydrogen that have been shuffled through the whole metabolic process. As we oxidize them, they're releasing that electron. That electron is going to move through an electron transport chain, and it's moving towards electronegative oxygen. So the oxygen you breathe is driving this process. Once that electron reaches oxygen, because glucose had a lot of hydrogens in it, there's hydrogens available. That electron that's been traveling through the electron transport chain is going to join with oxygen, and that's going to form a water molecule. That's why water is reduced. Water now has that electron that was contained in an electron transport carrier. Remember, like I said on Tuesday, all the oxygen you breathe comes from a water molecule being oxidized in photosynthesis, and the oxygen you breathe becomes a reduced water molecule. If biology does not amaze you, I don't know what else will. That is amazing that two completely different organisms exchange inorganic molecules like that. And we see that time and time again with these elements that we all share of life. Okay. Somebody's not muted. Somebody's going to mute themselves. I'll manage it. Okay, so questions on that right now before we go to draw it? Okay, so one thing I want to talk about before we draw is phosphorylation. Oh no, I gotta remember how to spell it. This is what panics me in life, is spelling things in front of you. I have all my little cheat sheets today so I can spell correctly. If I never had to spell, I'd be just fine. Okay, so phosphorylation is adding that third phosphate onto ATP. So it's taking ADP plus that free inorganic phosphate that's just there, because once you release that energy that was contained in this phosphate being bonded, they hang out together, is the nice non-chemical way to explain that. So phosphorylation is adding this onto this molecule to produce ATP. Remember, ATP is the big money maker. It's triphosphate. It has the three phosphates. Oh, I just inhaled funny. Excuse me. So ADP plus that free inorganic phosphate, this is disorganization, right? We're not going to get to ATP without an input of energy. So we need an input of energy. The phosphorylation is going to happen via chemoosmosis in what enzyme? What's the enzyme name? ATP synthase is going to catalyze this reaction. Yep, you got it, Jesse. ATP synthase. So we need an input of energy. The input of energy is going to come via chemoosmosis. And the chemoosmosis is to create those hydrogen protons that just flow in a passive transport way from high to low concentration. Those hydrogen protons have energy. Now, we're going to get these hydrogen protons in a variety of ways. So when we're doing photosynthesis, it's photophosphorylation. That means the energy to be transferred eventually into creating this dynamic of the high to low concentration difference of hydrogens comes from photons of light. We can have oxidative, phosphorylation. And this comes from reduced molecules being oxidized. This happens, right, in cellular respiration. Photophosphorylation is photosynthesis. So what do these have in common? Molecules that are reduced, that have the energy, become oxidized. That powers those proton pumps to pump hydrogens against their concentration gradient so that there's a high concentration of hydrogen in a specific space that then can passively flow through ATP synthase to provide the energy to do the phosphorylation. Photophosphorylation and oxidative phosphorylation, they have lots of energy associated with them. Okay, they can produce a lot of energy. They can transfer a lot of energy. They're kind of the big workhorses. We have one more type in cellular respiration that we talked about, and that's substrate level. We talked about this in cellular respiration. This is a substrate breaking apart. So it's a molecule breaking apart. As that molecule breaks apart, it releases a little bit of energy, and that little bit of energy is what's powering the ADP to become ATP again. It's releasing it, and it's providing a little bit of energy that way. This is energetically low. And the example, there's a couple, right? We see this when glucose becomes pyruvate, and we see this when acetyl-CoA breaks apart fully. So glucose to pyruvate creates this dynamic. You just get a little bit of ATP made, because that's not energetically very rich. So when we think about connections, photophosphorylation and oxidative phosphorylation are creating the same situation, right? It's powering a proton pump so that we can pump hydrogens against their concentration gradients. Electrons are providing the energy for those pumps. Then we have a glut of hydrogens in a specific space, just depending on what organelle we're in. And then chemoosmosis can occur. We can have those hydrogens passively flow through ATP synthase, which is going to catalyze the reaction, and the hydrogens will provide the energy input to take ADP and make it ATP. Questions on that? Okay. So let's just look then at the fill in the blank ones. So I debated, right, what I wanted to do. If I wanted to draw it out myself, I feel like, hang on. There it is. I feel like if I do it, it won't be as neat. And so if we do it in this capacity, I think it will be more helpful to you because it'll be a little less, it'll be less messy. So let me show you what I would do if I was you. That's really small. I hate how small it is. I don't like how when I make it bigger here, it doesn't make it bigger for you. All my thinking out louds. So what I would do to draw this out is this. Let me show you real quick. So if I was on the whiteboard, what I would do, maybe if I sit down, maybe I can draw it neater. Okay, so I would draw this, and this is a thylakoid. Okay, I would maybe draw it a little bigger, quite honestly. Okay, and it's really helpful. This is almost like a study moment. So if you think of my whiteboard as if you think of the whiteboard, that's a good question, Megan. So is it randomly cutting out for other people, the sound in the video, or is it everybody? Could be, I know, this is the eternal question. Is it your Wi-Fi or is it not? So that's a yeah. Okay. Let me see if it might be so with the new tablet, I have to run us a little differently than I used to, and that's going to make us echoey. So that's going to drive me nuts because I can hear it. You guys can hear that echo too, right? That's true. So I'm running us differently because if you've been in class with me, right, there's like 7,000 screens now in front of me. So AV is working on a way for me not to have to do it in this kind of disjointed way. But it might be just what it is. What if I move this closer? Maybe that will be helpful. It's staticky for me still. Oh, it's worse. Oh, well, that's good to know. Maybe if I go further away. Okay, is that better? But now I feel like I'm so quiet. This is like watching paint dry, isn't it? Oh, one day this will all be over, right? Oh, everyone's leaving. No. What? No. Okay. So my microphone, I'm running through my computer because we're signed in on this too. So I can go back to my fat little finger. Yeah, I think that's it. It's bearable. Okay, this is what we're gonna do. Give me a moment. You just lost the ability for my pretty little tablet though. Okay. That might make it better because I've taken my tablet out of play. Might make it better if I'm on that. Okay. Let's see. Still the same. Awesome sauce. No, it didn't. Awesome. That made it much worse for me. Oh my gosh. Okay. Well. Shoot. Can you come sign back into your Blackboard? Thanks. Okay, let me plug my tablet back in. Bear with me. I'll have you sign back in because, of course, I X'd out. Because why would I not have X'd out? Oh, maybe it's in there. Oh, good. Never mind. Thanks. Yeah, that's what I'm gonna have to do, Hannah. You're totally right. I'm just gonna have to keep going, and we'll figure it out after class. They should have given us, instead of like teaching us how to teach online, they should have just taught us how to work AV. Is really what they should have done this summer. Okay, so let's try again. Thanks, guys, for being patient. It's gonna make for a great YouTube video. So, I would try to draw it out. Okay, so here is our thylakoid. Okay, the whole whiteboard, if you imagine, this whole thing is the chloroplast. Okay, so and then what we'll have is this is the stroma, and in here this is the thylakoid. Okay. And this will allow us to kind of flow through everything. I'm going to erase it, make it a little bit bigger, because why not now make it just what I want. So I'm going to attempt to draw it out because I think that will be more worthwhile. So if we start in the thylakoid, right? This is the inner thylakoid, and it has a high hydrogen concentration. In my stroma, we have a low hydrogen concentration. Now remember, there's a ton of thylakoids. So this is happening across the whole thing, lots and lots of thylakoids. Oh, I can change colors on this too. I've just gotten excited again. Okay, so we have proteins embedded in the membrane, right? So we have photosystem 1 or photosystem 2. Remember, I'll never ask you that because it's really hard not to want to say that that's 1. We have our cytochrome complex, and I'll label these in a second. And then we have photosystem 1. So this guy, right, is, I don't know where I want to write it, PS and our PS2. That's the abbreviation for photosystem. This is PS1. And then this is the cytochrome complex. Now, there's two little accessory proteins that you don't need to know, but they're here and here, and they kind of help guide that electron transport chain. Now, within our photosystems, oh, wait, let's keep drawing our proteins that are embedded. How about that? Over here, we have an accessory pigment, and then we have our big peripheral protein, which is called NADP or NADP plus reductase. It's a specific enzymatic protein. Now down here, we have ATP synthase. And I always draw it kind of like a bottle like this. It's a little easier than the way that the book tends to draw it. But this is ATP synthase. Now remember, this is the thylakoid. This is a bilipid phospholipid layer. There's a bunch of them all stacked up. Now, in our photosystems, we have chlorophylls. Chlorophyll is the pigment that's going to absorb the sunlight. And so you have all these chlorophylls, and then you have like a special set of chlorophyll molecules that sit there and kind of absorb all that energy. And only sunlight that can be absorbed is used by photosynthesis. Sunlight comes in, right, and the chlorophylls are going to absorb that. These are photons of light. That's why it's called photosynthesis. So this is the light-dependent reaction because it's dependent on sunlight. So we have our water molecule because our plants love a lot of water in their cells. So our water molecule is just there, and it's going to get split. Okay, sunlight's going to help to do that. And what we lose is oxygen is a byproduct, it's called, or it's released, or it's a waste product, right? It's not used directly for photosynthesis. What stays in is all these hydrogens, right? That's how this high concentration of hydrogen in here. We've released an electron, and the electron is going to move into that chlorophyll molecule, the specialized ones kind of in the middle of the photosystems. And the energy that was transferred from these photons is going to help to power to push that electron to increase its potential energy. Now that electron has more energy than it once did. It didn't just magically get that energy, it was transferred from the photons of sunlight. Remember, specifically, plants super love the wavelengths of light associated with red and blue. Green is not used very much because that's the color we see, so that's reflective. So now we're going to go through an electron transport chain. So the electron transport chain I'm going to make yellow because it is in the diagram, but it also denotes energy. Anytime we see yellow, that's denoting energy. What's moving on this electron transport chain is the electron. Remember, electrons have energy. So as it moves down this chain, it's releasing the energy as it's moving down this chain. That electron, as it passes through the cytochrome complex, provides the energy for the cytochrome complex to pump hydrogens against its concentration gradient. So there's a few hydrogens out here, and they're able to be pumped in against their concentration gradient via the cytochrome complex because the electrons are going down in the electron transport chain. The electrons power that pump. So that continues to provide this huge concentration of hydrogens in here. And then that keeps it a low concentration out in the stroma. So the concentration outside the stroma remains low. Inside of the thylakoid, it remains high. Now, remember, photophosphorylation. We've reached it. Okay, we're going to take ADP plus the free inorganic phosphate, and it's going to become ATP. This is not for free. Energy needs to be available to do this. Where's the energy going to come from? It's going to come from hydrogens going through ATP synthase via chemoosmosis, and that's a passive transport then. Anytime you see the word osmosis, it's a passive transport. Why is it chemo? Well, because hydrogen's an element in water, and they just wanted to confuse the heck out of you. I don't know. It's not the world's best name. Right? And now we've transferred the photons of light into ATP, which is an energetic molecule. But wait, there's more. Right? We have more sunlight coming in to our photosystem too. Those photons of light will ping between the chlorophyll molecules as well. They'll go to the special chlorophyll molecules, and the electron from the electron transport chain is going to get there too. That energy is going to absorb into that, and it's going to ping this electron up to a higher energy state. It's transferring the energy in that sunlight to that electron and increasing the electron's energy level. So now this electron has a lot of energy, and it's going to go through another ETC, another electron transport chain. As it flows through that electron transport chain, we're going to then put it into NADP plus, which is the oxidized, and that electron is going to then facilitate NADPH. Now it has the energy, and those H's are out in the stroma. The electron facilitates that coming in, holding that there. We've reduced how positive that molecule is, and now it has the energy because it has an electron. And now we've completely finished our transfer of sunlight into these two energetic molecules. That drawing's not too bad. It's no worse than how I would have drawn it with a pen and paper. Not an art major. So now these both are going to then become hydrolyzed and oxidized to provide the energy to reduce carbon dioxide into G3P. So we've got the energy, we just need to harvest the energy. And that's going to go into the light independent reactions, or the synthesis part, or the Kelvin cycle. All of those things are the same thing. Questions on that? Because I'll have to erase it to draw the Kelvin cycle. I hope you agree that drawing it out like this slowly really does help. I think just to physically like follow the stuff is different than just watching me draw on an already drawn thing. It really is. Those cognitive psychology people are right, you know. I mean, when you write it down, it's a totally different connection to your understanding than it is just looking at it. Yeah, that's exciting, Hannah. Okay, so let's look now. Now we've got our energy. Okay, so we have energy. Wow, that got away from me. Energy. And so we've got NADPH and we have ATP. These guys both came from the light-dependent reactions, or the photo part. Now we're going to use this energy to reduce carbon dioxide. We're going to take an inorganic molecule and we're going to make it into a sugar. I mean, plants are amazing that they do this. And so this is going to help happen in the Kelvin cycle. We're in the stroma still, okay? The thylakoid is back this way, right, if we're looking in the directional of the chloroplast molecule. And our first step is carbon dioxide. We're going to have 3 of them go through to make one G3P, because G3P has 3 carbons in it. So we're going to need 3 individual carbon dioxides to make this sugar. So the first step is that carbon fixation. So carbon needs to come into the cycle. It needs to get fixed into the cycle. And we're going to have Rubisco help us catalyze that. Okay, so that's our CO2 acceptor. I'm like, I cannot think of the word. Okay. So we've got carbon dioxides coming on in. Now we need to reduce those carbon dioxides. So when we reduce it, we are increasing the energy. So we are essentially taking 3 inorganic carbon dioxides and we are reducing them into 1 G3P, which is a 3-carbon sugar. That's not going to happen without an input of energy. We are increasing the energy. So when we reduce, we're going to have ATP be hydrolyzed into ADP plus free inorganic phosphate. That's going to provide the exergonic reaction to release the energy to help in the reduction of these molecules right here. And then we're going to have NADPH, which is reduced. We're going to have it be oxidized. When it becomes oxidized, it's releasing energy. Where is that energy going? It's being applied to reduce, right? This is where energy needs to be applied. Oh, that got messy. Okay, that energy needs to be applied. These 3 are carbon dioxides aren't just going to bond to themselves. So when ATP is hydrolyzed and NADPH is oxidized, that's releasing the energy that's needed for this endergonic reaction of the reduction of carbon dioxide. So the output is G3P. It's 1 single G3P, and then not long after, it's going to become our good friend glucose. Now, on the chart, right, we've got all these 3 times 5x, 5 times 3x. That's just showing you that, there's constantly these molecules kind of changing form. And it's all being mitigated by ATP being hydrolyzed and NADPH becoming reduced. And so the last thing we need to do is we need to regenerate our carbon acceptor. Okay, and this needs a little energy input because we have to actually increase the amount of carbons because we just lost 3 carbons. So G3P is part of a larger molecule, and then 3 carbons come off of it. So we need to build it back up again, and so when we do that, we need some energy. So we're going to have, again, ATP will become hydrolyzed and become ADP, and we'll have NADPH, and it will become oxidized and become NADP+. That's going to regenerate, and it's regenerating Rubisco to then do it again. That's why it's called a cycle. Now, everything that's been hydrolyzed or oxidized is now going to go back, right? All those products are heading back to then be filled again with energy, to be reduced, and then to have a chemoosmosis. Yes, G3P has energy. This has energy. It's a simple sugar. Yep, has the energy. It's what carbon dioxides get reduced into at first. Just think of it as like a half of a glucose. It's not exactly that, but. Okay, then we have cellular respiration. What questions do you have on the Kelvin cycle? So the Kelvin cycle is the last stage of photosynthesis. Yes, the Kelvin cycle is the last stage of photosynthesis. Okay, let's draw out cellular respiration. Not whatever, but you know what I mean. Cellular respiration. We're going to draw it in kind of an alternative way, and then I'll show you where we're at in the PowerPoint. Okay, because I have, I can't show my little friends at home this because you don't have me dually, I don't think. But I have the blank pictures in front of me, but I have another picture that I really like to draw in terms of cellular respiration that will be very much like the photosynthesis one we just drew, kind of the same idea. So I'm going to draw the mitochondria, but I'm going to draw the mitochondria as a square. Okay. Okay, that is my mitochondria, and that is the outer membrane. Then we have that inner membrane, right? I'm going to give myself lots of space to draw and hope that I can do this in a way that is sensible. Okay, so this is my inner membrane. This is the cytosol. And in here, this is the matrix. In the middle is the matrix. Trying to think where I want to write it so it won't be in my way. I'll write it up here. This is the matrix. That is one beautiful mitochondria. So if you like videos, what I like about this picture is this came from Bozeman Science. So like Bozeman, like the town in Montana, it's where he lives. And so I just love how he draws it, and it just looks different than our book. But I think seeing it all together, seeing all the phases of cellular respiration together is really nice. And when your book does it, it's a lot. So I think it's nice to draw it like this, okay? So let's start first. We have glycolysis. Right? So when we have an olysis, it's breaking something apart. Glycosidic linkage, breaking apart. Glucose is breaking apart. Okay, so remember, glucose is a 6-carbon molecule. I'm going to write 6C so we can kind of follow them. And we break the glucose apart, and we get 2 pyruvates. So we get 2 pyruvates. That's why I drew 2 little lines there. We'll only follow one. And remember, pyruvates are 3 carbons. As we do that, we are going to reduce and oxidize NAD. So we have different electron transport carriers. So here's my oxidize, and we reduce an NADH. That NADH is going to shuffle its electrons to the last step, and we'll draw that in a minute. And remember, we make some substrate level ATP here. Just a little bit, just a couple. Now my pyruvate is going to, via passive transport, come into the mitochondria. But it's going to do so in a specific transport protein. And this is called 2 things. It's officially called, so if we think this is step 1, this is step 2. So this is pyruvate oxidation. So we're going to oxidize pyruvate. We're going to break it apart. And it's going to do so in this transport protein called the pyruvate dehydrogenase complex. So it's a specific enzyme. As pyruvate passively diffuses, we're going to take NAD plus, and we're going to reduce into an NADH, and that actually serves as a cofactor. It's going to activate that particular enzyme. Coenzyme A is a enzyme, it's a vitamin that comes in to help this reaction, right? So these are activating the pyruvate dehydrogenase enzyme to facilitate pyruvates being broken apart, or changing form, I should say better, into the substrate acetyl-CoA. Why do we spell acetyl wrong? Okay, acetyl-CoA is 2 carbons. So we've lost a carbon. Where does that carbon go? It is lost as a waste product or a byproduct, right? So we're losing carbon dioxide as pyruvates passively diffuse. Oh, you can't see the little cross in my A. There it is. As the pyruvates passively diffuse through the dehydrogenase complex. Now I've got 2 more carbons to deal with, right? I'm harvesting the energy all along the way. Here's an electron transport carrier. Here's an electron transport carrier. They have the energy. So acetyl-CoA is going to go through another beautiful little cycle, and this is the citric acid cycle, or the CAC. The CAC is happening in the matrix. Acetyl-CoA is going to join the cycle by joining with oxaloacetate. And then it's just going to finish breaking down. As we finish breaking it down, we are going to release the rest of the carbon dioxide. So what are we breaking down? That 2-carbon molecule of acetyl-CoA. Once it joins in via joining in with oxaloacetate, it's just going to cyclically change forms until it's broken down. And we're going to take NAD plus, and we're going to reduce the energy into it from that broken apart acetyl-CoA. and we're going to take our new little guy, FAD, and we're going to reduce it into FADH2. It's just another electron transport carrier. That's all that FADH2 is. It's a reduced molecule. Anything that has an H has the electron. It's been reduced. It has the energy. So now, all we have left is the energy that was originally contained in glucose. We've harvested all the energy. And we've done so by breaking apart the bonds. We put them into electron transport carriers. These guys are shuffling them down to here. They all are moving down to the last part, which is part 4. Okay, so the citric acid cycle is part 3. Part 4 is oxidative phosphorylation. Remember, there's 2 parts. We have an electron transport chain, and we have chemoosmosis. And I'm actually going to draw it up and down because I think it makes a little bit more sense. I hope so, at least. Chemoosmosis. Yee. So what we need to draw first is we're going to have proteins embedded in the inner membrane. And there's essentially 3, right? These are kind of what's going to facilitate. These become our proton pumps. So my electron transport carriers are coming to this. Now, in my matrix, I have low hydrogen concentration. In my inner membrane space, I have a high hydrogen concentration. At the bottom here, with the top of the little vase sticking into the inner membrane space, I have ATP synthase. So now I have NADH and FADH2. These are my reduced molecules. Now there's many of these. There's not just one of each. There's several of these. Okay, so keep that in mind just for a moment. There is a little protein here, just for say being accurate, I guess. So as NADH becomes oxidized, right? So we have NADH and it becomes oxidized to NAD plus, right? That's messy, sorry. It releases that electron. I'm going to go to a yellow color in just a second. That electron is going to go along an electron transport chain. It's falling down the hill energetically. So we've got this electron moving through an electron transport chain just like we did prior. Right? What is it falling down to? What's driving this? It's our electronegative oxygen down here at the bottom. This electron is attracted to that oxygen molecule, so it's going to come here. Now, the reason why we continue to have a low hydrogen concentration in the matrix is because any hydrogens that are around here are eventually going to, with the help of this electron, bond with oxygen, and we reduce it into water. So the oxygen you breathe becomes water with the help of this electron flowing into it. And then the water leaves. Now this little protein right here, this is the one that is where FADH comes in. So when this is oxidized to FAD, it releases this electron here, and it joins the electron party. It's like, woo, yeah. Okay, then you have NADH here becoming reduced. So this is happening at all these sites, contributing their electrons to their electron transport chain. So there's a lot going on energetically. So what are these electrons powering? They're powering, just like they did prior, hydrogens to pump against their concentration gradients. so that then we have a high concentration of hydrogen in the inner membrane space, and we have a low concentration of hydrogen in the matrix. Two things are doing that. When we release that electron, these hydrogens are being released from those reduced molecules. They're getting pumped out, or they're joining in oxygen to form water. That's what maintains that low concentration gradient. Remember, we want to create that electrochemical gradient, that membrane potential. So you got all these hydrogens in the inner membrane space. That creates the dynamic then for chemoosmosis. Chemoosmosis is going to flow, or the hydrogens via chemoosmosis are going to flow through ATP synthase, which then is going to take ADP plus the free inorganic phosphate and create ATP, right? That's what's coming out of the ATP synthase. It's catalyzing that reaction. The hydrogen protons are providing the energy that's needed for that endergonic reaction. ATP synthase is catalyzing it for us. It's making it happen faster, and it's lowering that activation energy. And then, like magic, voila, woo, tons of energy, right? This is oxidative phosphorylation, and this is where the majority of our energy comes. Not from the substrate level down here at glycolysis or after the citric acid cycle. Sorry, I forgot to mention we get a little substrate level ATP2 there. Most of the energy that you're making is coming from this process right here. When we oxidize those molecules that were reduced, they were reduced from the various metabolic reactions that were breaking apart glucose. So that's really messy, but hopefully that flow helped as well to trace it all along. Questions on that? Yes. Yeah, I'll go over step 2 and 3 again. I'm going to show you on the picture. What's the difference between intermembrane and inner membrane? I've seen both in content. I don't think there's a difference. I'm pretty sure I probably said both. I tend to say inner membrane, but intermembrane would be the same thing. It's the space right, oh, you can't see me right there. It's the space right here in between the inner and the outer membrane of the matrix. I assume that's the same. Okay, let me show it to you on the PowerPoints. Which process occurs inside the matrix? It's the citric acid cycle. So let's look at it again. Not that one, not that one, this one. So here are the pictures that are blank, right? So you can draw on these too if you want. So if we're here, we have pyruvate going through. The matrix is in here, and this is where the CAC is going to occur, the citric acid cycle, right? It's the cycle. We started with glucose. So to get to pyruvate, this needs to break apart further, and so it's going to come into the mitochondria, and it's going to do so like this, right? So here's our transport protein. Here's pyruvate. It's got carbon. It's passively going through the phospholipid bilayer, but it needs a transport protein because it's too big. When it does that, pyruvate is the substrate, and it's going to go through a specific reaction. That specific reaction is to yield the product acetyl-CoA. So here's acetyl-CoA. And so this transport protein is the dehydrogenase complex. And this is pyruvate oxidation. We're oxidizing pyruvate. So as we do that, carbon dioxide comes out because it has to change shape. That's a reaction. There has to be some sort of a change. To have this enzyme activate and actually function, we need to reduce this molecule. So NAD comes in, causes the activation of the enzyme, and we have NADH. Then, as a result, that's our reduced molecule. It has the energy. And then coenzyme A comes in as the other activation, which then actually ends up bonding inside the acetyl-CoA. That's why it has this CoA name. Then acetyl-CoA is going to join into the citric acid cycle, which is in the matrix, like this space really is. This is the matrix. And the citric acid cycle will fully then break apart these final two carbons. Acetyl-CoA, this CoA part, this is what's going to join in with oxaloacetate. Like if we're thinking about how it bonds in, it needs to clear together, and it's going to do so with that facilitation of that small little coenzyme there. And then acetyl-CoA and oxaloacetate becomes citrate, which is citric acid, and that's why it's called the citric acid cycle. And so this is happening in the matrix. So then, if you look, here's my acetyl-CoA. And so this is what we've drawn, right? It's just the book's depiction of it. So this 2-carbon is joining in here with oxaloacetate. And on the side here, it has the keywords that you should label. And then we're going to finish breaking it down. As we finish breaking it down, we release those last 2 carbons because we're not really after the carbons necessarily. We're harvesting the energy that's contained between those carbon bonds. And so we take oxidized NAD and we reduce it down to NADH. We make a little substrate level ATP, just a little, tiny bit. And then we take reduced NAD or FAD and make it FAD2. And then NAD and make it NADH as well. Then we've harvested all the energy. So now all these reduced molecules are going to go to the oxidative phosphorylation where they're going to become reduced, which is going to power the electron transport chain that's going to power the pumps that's going to pump hydrogens against their concentration gradient into the inner membrane space, which creates a chemoosmosis, flow through ATP synthase, which then allows the hydrogen protons to provide the endoganic energy needed to take ADP and make it into ATP. You do have access to those diagrams for the fill in the blanks. Yep, these are all on Blackboard underneath exam 2. Yep. The filled in ones are there and the blank ones are there. Okay, so questions. More questions. I love questions. I miss crazy questions that you would ask me. Hopefully this helped, right? Because we just go through it once and you can't know what you don't know until we go through it a couple more times. And so hopefully this drawing it out and slowing it down is really helping you see the processes a little bit more so that you feel ready to take the exam. Okay. That is the first lecture I've ever sat in like 10 years. It was really weird. Okay, so if you guys don't have any questions, then just let me know. You know, if you have all day tomorrow to take the exam, I'm around if you want to shoot me an email with questions, or we can jump in a Zoom together with last minute stuff. You know, I'm here to help with anything I can. So have a good weekend. Enjoy.